All right, guys, we are going to do a small group review over in your STAAR book over cell cycle and meiosis, okay, and kind of the difference between the two. So if you could have your STAAR book out, um, have your STAAR book out, and turn to page 17, and then fill in the chart and the diagram on page 18. Okay, and if you have time, answer those questions. Number 4, page 24. Number 7, page 25. Number 10, number 14. So if you want to hit pause, that way you guys can try this video out by yourself, and just try to do it by yourself before I go into it. Okay, it's all about cell cycle this time, so try to remember all the phases, what happens in each phase, all of those things. Okay, so if you want to press pause now, I'm going to move on. So this is what the cell cycle looks like. Okay, we start with interphase. Interphase is where you have it's the longest phase, and that's where you have the growth and kind of the preparing to divide. Okay, you have G1 in there, G1 and G2. This is where the cell grows and kind of gets ready to split into two. And this is where you have DNA replication S phase, which is a very, very important phase. Okay, you need to double the amount of DNA, and if that doesn't happen, then the cell cannot duplicate or have normal duplicated cells. Okay, if you notice, it looks like a bowl of spaghetti. Okay, because that's how much DNA we're getting. Once it's ready to go, we are going to have prophase. Okay, and this is when this is the first phase of mitosis. This is when the DNA winds up and condenses into chromosomes. Okay, so if you remember the hand signals I taught some of you guys, your biology teacher might have taught you guys, and that's when the DNA condenses into chromosomes, which looks like X's. Okay, next we have metaphase, and this is where the chromosomes meet in the middle. Okay, so they meet in the middle, they're getting ready to divide. They also have these spindle fibers that kind of lasso to each one, and they're getting ready to pull. Okay, next one, anaphase. Chromosomes separate, so this is where the spindle fibers actually pull it apart. Okay. Next one, telophase, last phase of mitosis, and this is where the DNA is on opposite sides. Okay, both sides of the cell. It's still one cell. It's not two cells yet. It's one cell. And in cytokinesis, you can't really see it because of my face. Cytokinesis is the last one, and this is where they divide. Okay. So you have all of these phases to make sure that they're making sure that everything is going right. Okay. But the number one phase that's super important is DNA replication, and that happens during S phase. So this process takes place during the S phase of interphase. It's necessary for mitosis and cytokinesis to divide, and that way you have two identical cells. Okay. If you don't have two identical cells, like all of your skin cells, then it won't work. Okay, that's when bad stuff starts to happen. Okay, so remember all of these phases. Now that we know all that, look at your page 18, that chart that I have right here, and that little cell cycle sort of diagram, and just check your answers for the first one. The first one is, First one is C, chromosomes condense. Okay, nuclear envelope starts to break down, so the nucleus kind of starts to disappear, and this would be prophase. Okay, next one is D, because that's when the chromosomes line up at the equator, so this would be metaphase. Okay, next one is A, spindle fibers start to pull the chromosomes apart, and last one for mitosis is B, telophase, because that's the final stage. That's when you have two nucleuses form. Okay, and if you just want to check to see where you put, if you have all the right places, this whole part is interphase, G1, S phase, which is DNA replication, one of the most important ones, G2, and then when we start into mitosis, prophase, metaphase, anaphase, telophase, and the very last phase, cytokinesis. So just make sure to check that. If you have any questions, check back into page 17 and kind of check those answers again. Now turn to page, I think it was 24, for the STAAR questions to practice on. For number 4, the activities in the cell cycle occur during specific phases. In which phase of the cell cycle is DNA replicated? That's what we've been talking about the whole time, right? So we have mitosis, G1 phase, G2 phase, and S phase, okay? I don't think I talked about it in that last slide. The S stands for synthesis. Synthesis means to make, so we're making DNA. Next one. The model of the cell cycle includes two arrows that each represent a process in the cell cycle. What do the two arrows represent? So we look at arrow 1. Arrow 1 is the longer part of the cell cycle. We have arrow 2, kind of looks quick, okay? So we always know that preparing to divide takes a lot longer, making sure that everything's correct, all these checkpoints, everything, and the actual division doesn't take that long. So what do the two arrows represent? The only one it could be is arrow 1 is interphase, arrow 2 is mitosis, okay? Interphase always takes longer, maybe hours, days, depending on what type of cell it is. Okay, part of the cell cycle is shown in this simplified model. This part of the cell cycle is best described as the process that A, duplicates the DNA, B, completes cell division, C, maintains genetic homeostasis, or D, synthesizes new molecules. Okay, so there's a couple of that look correct, but since we have two cells right here, it looks like they're splitting, and they finally split. This is the end of cell division. This is when it actually divides. Okay, so we kind of go from telophase all the way to cytokinesis. For number 14, people who have leukemia, a cancer that affects white blood cells, are often given cytarabine. This drug inhibits the synthesis of DNA. Which phase of the cell cycle is most affected by cytarabine? This word, I know it confuses a lot of you guys. Inhibits means to stop. So it stops the synthesis of DNA. Which phase of the cycle would be most affected? G1, S phase, G2, M phase. Only one it could be S phase or synthesis phase, making DNA. Okay, so for this next part, I want you guys to read, highlight, brainstorm page 51. And then there's some questions at the bottom with the little question mark. Answer those 4 questions. Fill in the chart below that compares and contrasts on page 23. And you're comparing that with meiosis, which is on page 51. So if you guys want to use those 2 pages to fill in that chart, then you're going to answer questions number 7 on page 55 and number 16 on page 58. And then on page 21, just answer the question on page 22. Kind of read page 21 and answer page 22. So if you want to hit pause before I get into it, you can. Just try it by yourself, okay? So right here we have meiosis. Very, very similar to mitosis, except this is when you have, we're trying to make haploid gametes. So in mitosis, we're trying to make identical cells. In this one, we're trying to make cells with half the amount of DNA, and we're trying to make sex cells, okay? This is going to be important because during sexual reproduction, when the sperm and the egg come together, it makes a full baby, okay? So this is the process. It goes through meiosis 1, meiosis 2. What's going to be really important is crossing over, which happens right here, kind of around here, in prophase 1. Crossing over is so that it increases genetic diversity, and because it combines different parts of the DNA. So it happens right here, because you see that the red and the blue switch, and it keeps going, and we end up with 4 gametes that are completely different. This is why you and your brothers and sisters look completely different. Okay, so make sure that you know crossing over happens in prophase 1, and it's to increase genetic diversity. So diversity just means differences. So this is where crossing over happens. So if you could flip to this page right here, where we talk about meiosis, and it just says, what is the importance of crossing over? Genetic diversity, okay? For number 2, what cells go through meiosis? We have gametes. So somatic cells or body cells are going to be from mitosis. If your cells couldn't go through meiosis, how could this affect you? Well, if you have no meiosis, you have no gametes, so you have no egg or sperm, so you can't reproduce, okay? And This could affect many organisms who want to get more traits out there, more genetic diversity out there, and it could affect an ecosystem. Okay, so meiosis needs to happen. And this is the last one. We set it up so that you guys could see the specific differences. Okay, you could do it any way that you want, but for type of cell that is made between meiosis and mitosis makes body or somatic cells. Meiosis makes gametes or sex cells like egg or sperm. For number of cells, mitosis makes 2. Okay, meiosis makes 4. Mitosis makes genetically identical, completely the same. All your skin cells look the same. All your brain cells look the same. Meiosis, they all have to look different because of that crossing over. Mitosis makes diploid, so the normal amount of DNA. Meiosis makes haploid, half the amount of DNA. So haploid, half. If you could turn to these pages right here, for number 7, I think it was page 55. In the early 1900s, Thomas Hunt Morgan was among the first scientists to contribute the chromosome theory of heredity. Morgan's investigations into heredity in fruit flies led him to propose that the event represented in the diagram sometimes occurs. Which statement about the event represented in the diagram is valid? So what is going on in that diagram? You have 2 chromosomes right here. They mix up kind of, and then they're like completely different. So what is going on? Well, based on what we know, this kind of looks like crossing over, but let's read the questions or the answers. The event represents RNA translation. That already sounds wrong. That sounds like protein synthesis. The event takes place in bacterial cells. Well, we don't really know that. It doesn't really say anything about that. The event produces genetically identical daughter cells. Well, that sounds more like mitosis, and crossing over happens in meiosis. This event provides genetic diversity in eukaryotic cells. That would be it. Which diagram demonstrates, for number 16, which diagram demonstrates how crossing over contributes to genetic variety during meiosis? So which of these is talking about genetic variety, genetic diversity, crossing over? Okay, so we're going to look for which one looks like crossing over. Looks like this one. I know more people are going to pick this one because of new strands, daughter molecule. Don't get confused. This is DNA replication. This is crossing over. Remember, just a little piece kind of like switches. And then just a little quick information about cancer. This is on page 21. What is cancer? Here is a normal cell division. So if there's cell damage, the cell goes through apoptosis or cell suicide. It just kills itself, so that way it doesn't make more of that same bad cell. In cancer, the bad cells keep on reproducing, mutations keep on happening until you get an uncontrolled growth, keyword uncontrolled, and that eventually forms a tumor, okay? So uncontrolled, unregulated cell growth where we can't really control it. Here is a not real life example, but a more real life example. Cancer is caused by cells that are not controlled by the cell cycle. Many cancer medications interfere with the cell cycle to stop tumor growth. The model below shows the stages of the cell cycle at which 3 drugs interact with cancer cells. So these 3 drugs, drug A, B, C. Which drugs will prevent the cell from entering the cell division phase, but still allow DNA to replicate? Okay, so we don't want it entering the cell division phase, but we still want the DNA to replicate. We still want DNA in there. So the only one it could be looks like B, because we don't want it to go through mitosis, right? We don't want it to divide, but we still want the DNA to happen, the DNA to replicate. So this would be S phase right there. If you guys have any questions about any of this, I know it's a lot, you can ask your biology teacher, okay? Thank you.